Welcome friends, welcome to the Knowledge Club online. Now I am going to discuss about the pre-mRNA transcription, termination site and processing, means poly A. The 3' end of the mRNA genes has many regulatory elements which perform functions like stability of mRNA, elements for degradation of mRNA, transport to certain loci in the cell, elements that make mRNA as untranslated, making mRNA inactive for the storage. Also has site for the binding of siRNA for inactivating or degradation of mRNA and poly A addition elements. But transcription of mRNA is terminated beyond these elements. The termination site of transcription varies from one gene to the other. The distance from poly A site and termination site varies. Most of the pre-mRNAs are cut at the 3' region at specific site and poly A's are added. In some, such as histone, 3' processing is different, for they don't contain poly A tail, but one of the histone mRNA is added with a poly A tail, very unusual. Now, the addition of poly A tail and its importance. With the exception of histone mRNA, all other mRNAs contain a poly A tail. Poly A addition takes place while the 3' end of the pre-mRNA is produced on the template. Termination site or transcripts of structural genes is 1000 and odd nucleotides downstream of 3' translation terminated site. Termin TER. Transcriptional termination and polyadenylation event are not linked and they are independent events. While the transcription termination at 3' end provides facility for cutting at a particular sequence and polyadenylation, this happens while the transcription is at its end, perhaps even after termination. The enzyme components required one again are found assembled at a CTD tail of the large subunit of RNP2. The pre-mRNA at its 3' non-coding region contains certain sequences such as AAU, AAA at about 20 to 30. Now the sequences such as AAUAA at about 20 to 30 nucleotide from the poly A start of the functional mRNA. This sequence is considered as a poly A signal. From database of vertebrate mRNA, certain consensus sequence has been established. Any change in the sequence affected poly A addition. The sequences A98%, A86%, U98%, A98%, A95%, A96%. In addition to poly A signal at 24 to 34 nucleotides downstream from the poly A signal, another sequence GU and US are found in pre-mRNA. The GU and US rich sequence in mRNA is required for processing. Yeast cells does not have poly A signal sites, instead they have UA rich sequence in the upstream of poly A start site. Even plants have poly A signals such as AAU, AAA, but prokaryotes don't contain poly A sequence at their 3' ends. To perform polyadenylation, the following factors and enzymes are required. Polyadenylation can be inhibited by cordycepin, 3' deoxyadenosine. CPSF, cleavage and polyadenylation specific factor, is a complex of 4 subunits such as 160 kD, 100 kD, 70 kD and 30 kD each. The complex binds to AAU-AAA sequence. By doing so, it specifies a nearby position of poly A adenylation start site. This complex also interacts with poly A polymerase and cleavage stimulation factor CSTF. CSTF, cleavage stimulation factor, identifies and binds to GUNU rich site. This identifies the site for cleavage. This complex consists of 77, 64, 50 Kd protein subunits. This interacts with the cleavage factors such as CF1 and CF2. CF1 and CF2 cleavage factors CF1 and CF2 associates with CPSF and CSTF. The enzyme consists of 68 to 69, 29 Kd subunits. These are cleavage enzymes. PAP. The molecule's mass of poly A polymerase in 82 Kd a monomer. It is required for facilitating cleavage and catalyzing poly A addition. For its activity, it also requires poly A binding protein to PAB2. The enzyme is 689A long and contains N-terminal, RBD domain, RNA binding domain, PM domain, polymerase domain, and NLS1 and NLS2 sites for nuclear localization. They also have serine and threonine rich sites for phosphorylation. PAB2, PAB. This is not PAP. This is PAB2. This 33 Kd protein is the counterpart of cytoplasmic poly A binding factor PAP1, PAP1. The binding of this factor stimulates PAP's activity and also it regulates the length of poly A tail and provides safety from 3' exonuclease. Cleavage factors PFS1A, CSTF1B are involved in cleavage, but functions do not correspond exactly to the mammalian counterparts. Now, this is the addition of poly A tail and its importance. In this picture, shows the process of polyadenylation. Now, here the poly A adenylation components assemble at the site. The polyadenylation in yeast cells, yeast lacks a distinct UAA sequence, and polyadenylation is associated with a less well-defined AU-rich and A-rich sites. The CFPS equivalents are CFT1, CFT2, CFT2 and YTH1. YTH1, a factor comparable to CSTF. RNA 15, RNA 14, PFS2 bind RNA 15, RNA 14, PFS2 binds upstream of CPFS. A number of factors appear to be involved in binding downstream of the cleavage site as for mammalian CSTF. PEP1P is the poly A polymerase. Two hybrid screens have shown it to interact with a protein FIP1 FIP1P, which in turn bridges to the CSTF like factors RNA 14 and RNA 15. Which then binds to a protein complex associated with an addition upstream AU-rich sequence marker and HNRNP like protein HRP1 HIP1 binds in between CSTF and CPSF equivalent factors. CPSF73 as the 3' processing nucleus, the core phosphorylation machinery. RNAP2 is not included, is illustrated as it would assemble in a pre-cleavage complex on an mRNA precursor with CPSF73 at the site of cleavage. The AAU, AA and GU-rich signal sequence are indicated, and the arrow denotes the cleavage site. The protein-protein and protein-RNA interaction depicted are consistent with available data. This is the diagram illustrates the different steps in the addition of poly A tail. Now come to the mechanism. At the 3' end of the pre-mRNA is generated, which is generally 200 or more nucleotide from the poly A addition site. CPSF and CSTF bind to their respective sequence sites, that is poly A signal and GU or U-rich sequences respectively. These two interact in such a way the RNA in between is looped out and both CPSF and CSTF are brought together. This interaction also brings cleavage sites together. At the same time, CF1 and CF2 bind to CPSF and CSTF is such a way they can easily locate the cleaving site and have access to RNA for cleavage. At this point, binding on the PAP to the complex stimulates the cleavage factors to cut. Then, cleavage factors cut the RNA and CSTF with the rest of the 3' end of the RNA are released. Cleavage and poly A addition is simultaneous and it is so quick that no one has isolated mRNA with a poly A tail so far. For PAP to be active, it requires some stimulation from PAP2 and then it leads to the addition of adenine nucleotides one by one. There is no template for synthesis of poly A RNA strand as an extension. This enzyme behaves just like a DNA terminal nucleotide transferase. Initially, adenylation up to 10 to 12 nucleotides is slow and then poly A addition is fast. As more and more poly A are added, more number of PAP2 associate. When the length of the poly A tail reaches about 250 nucleotide size, it stops, but PAP2 remains bound to the poly A tail. In cytoplasm, the PAP2 is replaced with PAP1. Nuclear poly A tail is about 220 to 250 nucleotide long and cytoplasmic tails are less than this. Poly A tails present in eukaryotic mRNAs was discovered bidiurnal when the RNA was digested with either RNase A or RNase T1. What was left was only is RNase cut RNA after Cs and Us and RNase T1 digest nucleotides after Gs. Poly A addition is not inhibited by acetaminosin D, which inhibits DNA dependent RNA synthesis. There are no such stretches of poly Ts to 200 to 250 nucleotide long in DNA of a gene. Splicing of hnRNA starts even before poly A addition initiation. Poly A segment is subjected to constant turnover. There are many mRNA in which intron is used for polyadenylation. What is the function of poly A tail? It protects mRNA from 3' exonuclease digestion, for the tails are bound in PAP2 in the nucleus. It protects mRNA from 3' exonuclease digestion for the test bound by PAB2 in the nucleus or with PAB1 in cytoplasm. It provides stability to mRNA by interacting with a cap-bound factor and PAB-A-bound PAB1. This leads to circularization of translating mRNA. Poly-A RNA produces more polysomes than poly-A-less mRNA. Poly-A enhances translational efficiency for it is bound by CBP1 protein. Poly-A neighboring protein region is used in transporting and localization mRNAs in the cell interior. Microtubule fibers and actin filaments and motor proteins like dynein, kinesin, and myosin proteins are used as motor proteins to transport mRNA to a specific destination within the cell. Example, nanovascular and other mRNAs in developing Drosophila egg. In cytoplasm, nuclear PAB2 is replaced by cytoplasmic PAB1. It is believed that each time of mRNA is translated, its poly-A size gets reduced and it is called ticketing. But now it is known poly-A is degraded by a cytoplasmic enzyme called poly-A nuclease PAN, which sequentially released adenines. In the presence of PAB1, degradation is not pre-evented but slow till 20 As and then the degradation is blocked. Some poly-A RNAs are stored in cytoplasm as informosomes, which contains a short chain of 20 or odd poly-As. At the upstream of poly-A side chain sequences called CPE, cytoplasmic polyadenylation elements facilitate the binding of CPEB. Mask-in binds to CPEB and cap-bound EFG and 5' end, thus circularizes the mRNA. At the time of activation, the mRNAs are added with a full-length poly-As. Addition of cytoplasmic poly-A depends upon the sequence found at 3' UTR sequences upstream. At about 60 nucleotides from the first A of poly-A tail, which is called cytoplasmic phosphoadenylation element PEA, which this site is sensitive in the sense the presence of its facilitates the addition of poly-As. Substitution in this region is inhibitory to polyadenylation and promoted to deadenylation. The upstream of AAUAA sequences like AAUAA UUAU and CPE are very important for stored mRNA. CPE is used for polyadenylation in cytoplasm. It is only after polyadenylation the hitherto inactive mRNAs become active and they are then translated. This kind of activation has been observed in xenopus, oocyte and other systems. The AUUUUA rich RE sequences found in 3' UTR are regulatory sequences used for degradation of mRNAs. Similarly, there are antisense sequences for the binding of siRNA that leads to degradation of mRNA. When maturation specific synthetic mRNA and another mRNA without such sequences are added to an oocyte extract and maturation specific element containing mRNA gets polyadenylated but not the other. This UUAU is called cytoplasmic polyadenylation element. Poly-A addition to such mRNAs also require the presence of AAU-AAA signals. This lecture may be helpful to you. Thank you.